Tick Young, who's going to talk about ApoE in late.
Okay, thank you very much for the organizers for giving me the chance to share some of the works that we did before. So I'm going to talk about the notorious ApoE in neurodegeneration. So no conflict of interest. Okay, so here in this meeting, everyone is now aware that late NC is commonly comorbid with Alzheimer's disease neuropathological change. And when they're together, they're associated with faster decline, both in neurodegeneration and cognitive decline. So it's reasonable to wonder whether their coexistence is just a coincidence or is there some shared pathomechanisms. And it's very reasonable to wonder whether there's a shared genetic risk factor between these two very commonly comorbid conditions. And now we know that ApoE epsilon 4, a very strong genetic risk factor for Alzheimer's disease, is also a strong genetic risk factor for late NC. So this is a data from ROS-MEP cohort collaborating with Julie Schneider. And here, ApoE epsilon 4 was actually associated with higher latency stage by odds ratio of 2. This was actually even stronger than TMM106B haplotype that we just heard about, which had an odds ratio of 1.3 to 1.4 in our hands. And even after adjusting for quantitative burdens of amyloid beta and tau, we still saw the association going strong, both in the stages and also semi-quantitative latency burden across brain regions. And then we focused on a small subset of ROS-MEP participants in an updated data set. We had like 170 people who had no evidence of Alzheimer's disease pathology by immunohistochemistry. So we selected people without any evidence of amyloid beta accumulation per immunohistochemistry, and there was still an association between ApoE epsilon 4 and late NC. So this suggests a model where ApoE epsilon 4 is a truly pleiotropic locus, affecting both ADNC as well as late NC. It's not just simply going through Alzheimer's disease pathology. And so the relationship between these two pathologies are a little bit more complicated. And also the link between Alzheimer's disease and late are not fully explained by ApoE epsilon 4 either. So there's something else going on. And then we shifted our gears to look into the hippocampal sclerosis. And in ROS-MEP, ApoE epsilon 4 had an association with hippocampal sclerosis, and this was not explained by A beta or tau. But when we adjusted for latency in the model, the association disappeared, which suggests the model that's agreeing with what Pete Nelson has been saying multiple times today, that at least a big portion of hippocampal sclerosis is likely a downstream event of late NC instead of something unrelated, which is unlikely per statistics anyway. So our genetic studies show that latency is likely upstream event of hippocampal sclerosis. So to put things together, ApoE epsilon 4 goes through a portion of it seems to be going through Alzheimer's disease neuropathological change, especially amyloid beta, but there is a significant component that is unrelated to amyloid beta. And we did a brain transcriptome study, again using ROS-MEP data set of like about 420 older adults who had all the data necessary. And we created gene co-expression modules by grouping genes that had similar expression regulation patterns. And actually among the gene co-expression modules, a module that includes some key genes in lysosome and myelination pathways stood out as the one that's connecting amyloid beta and late NC. And even more interestingly, the TMM106B haplotype we just heard about also dysregulates lysosome myelination module that we just talked about in the same direction as amyloid beta does. So these analyses show that seemingly different pathways of ApoE A beta and TMM106B related pathways are nicely converging onto lysosomal pathway that later leads to late NC. Of course, this is a cross-sectional causal modeling from postmortem human data, so it should be interpreted cautiously, but we're excited to see that genetics guided us to actually look into this picture. And we now learn that it's from other fields and fields of late NC and neurodegeneration research in general, that genetic risk factors are not only could be useful for disease risk prediction, but also could be useful as a tool to identify further biological pathways that can be targeted for biomarker and therapeutic target discovery. So with that, I'll just summarize what I've just said. So ApoE4 is a very important genetic risk factor for latency. And this association is not fully explained by Alzheimer's disease neuropathology alone, and there's a strong suggestion of pleiotropic effect. And lysosomal dysregulation might be connecting ApoE, A beta pathway, and TMM106B pathway, leading to latency. But one major limitation of all the data that I showed you is that this is coming from pretty genetically similar population that like coincides with EUR reference panel, basically like European ancestry. And this association between ApoE4 and latency hasn't been clearly replicated in other ancestries. So further work is required to further dissect this very complex and important locus. So I'll conclude by thanking my collaborators and mentors, Dr. White, Diager, and Schneider, as well as my scientific and career mentor, Dr. Rita Sperling, and ROSMEP study participants and study staffs, and funding from NIH. Thank you.
Thank you very much. Genesis so far pretty quick. And so now we move on to Dr. Fardo talking about gaps and opportunities. So you can go slow there, Dr. Fardo.
I'll slow down. Get us some extra discussion time. Great. So thank you for the opportunity to participate in this workshop. This has been fantastic. As a statistical geneticist, biostatistician, I have learned a ton in the first little bit, and I'm looking forward to learning more. So I do get to talk about, and we'll test this out before you leave. Thank you. To talk about the gaps and opportunities in the genetics of late. One of the things we've seen a good deal today is that there are multiple comorbid pathologies, multiple comorbid proteinopathies. This graphic here shows a binary classification of tau, amyloid beta, alpha-synuclein, and TDP43, just the 2 to the 4th 16 combinations of yes or no's for each of these proteinopathies. And you can see from this convenient sample of a couple thousand people from NAC, ROSMAP, and ADNI that it's across the spectrum. You see a wide amount of variability in the comorbid pathologies. And this is important because if we're wanting to identify the genetic predisposers, the biological pathways that are involved in any of these proteinopathies, we need to be able to know what we're attacking, right? And so if we're interested in identifying tau, the idea that amyloid beta or alpha-synuclein could influence that as well has to be incorporated into the models explicitly that we're using to test these. And so how we can do this is a very simple binary yes or no classification. There's a lot more to it, right? That there's severity, there's importance. The paper that was the consensus staging paper is super important for that very reason. And in order to be able to address these again, we need those classifications. We need those rubrics, a paramount need. This might be the least controversial statement of the day. Limited sample size means low power. And we have limited genome-wide association study sample size. This was the hippocampal sclerosis GWAS that was presented a little bit earlier, and requested post hoc power curves based on the sample sizes that we had at the time that are not so different than what we have currently for late. And what you can see is the only things that we can detect are with sufficient power have high effect and are fairly common. The alleles are fairly common. So what that means is what we've seen thus far with the limited sample size is only a small selection of what we can find with more samples. And this has been fairly well known across multiple different diseases, multiple different disease phenotypes. We know that even among traits that have different genetic architectures, that the more sample we have, the more genetic variants that we can discover. And so this is just a couple different papers that show that explicitly. And we have, this is about the sample size we have for late. And we have sufficient power to detect big effects with fairly common variants. So a big gap is simply the sample size we have from autopsy studies. So that's a plea that I'm making to get more neuropath. This is a review article that was published last year. The context is Alzheimer's disease, GWAS. And here again, these phenotypes are clinically based, right? And so the arguments that are made in this review article that looks over the history of Alzheimer's disease, GWAS, which is a necessary step in discovery, is that neuropath leads to definitive risk loci. So if we're able to have a cleaner phenotype and understand what it is that we're looking at, the better chance we get those TMEMs and those granulins and those ABCC9s. We need more deeply phenotyped samples with pathological confirmation. And again, this is important because this clinical syndrome umbrella of sorts of Alzheimer's disease, dementia, is not necessarily the particular pathologies that we're interested in uncovering. Another call that has been mentioned in other talks is we very much need to recruit diverse ancestry. This little almost cartoon is just an example of one of the reasons why this is important. Many reasons why getting a diverse ancestry participation is important, but this is a very tangible one. This is a brain imaging phenotype with sample sizes similar to what we have for late. And if we only examined, which is fairly common, European ancestry individuals, this gene would have never been discovered because the quality control pipeline would have removed it from analysis because it's too infrequent. So if we only looked at European individuals, if we didn't have a diverse representation of participants in the study, this particular gene would not have been discovered. And so here's a very tangible example of that need. This paper here argues for including multiracial individuals as well. Very much agree. So a big gap is garnering additional participation for autopsy studies with diverse ancestry. This was a ubiquitous cartoon for a few years after it was published. This has been 15 years ago or so. And the idea here is if you've been at any human genetics meeting in that era, you saw this at about half the talks. This cartoon is saying we can identify, we can estimate heritability, the proportion of a disease phenotypes variants that can be explained by the variability in genetics. We can estimate that. But there was a gap of being able to explain that. And this was discussed earlier in the context of explaining dementia risk in general, a very similar concept. We've filled some of these gaps already in a wide spectrum of diseases, including Alzheimer's, including late. But we're now able to see a lot more. So there's reasons for that gap in heritability. What we know is there and what we've been able to explain. One of the things that we're able to do now is assay different types of variants. This paper by colleague Mark Ebert talks about rescuing variants that were not identifiable with short read sequencing, but are rescuable. So we're able to look at those variants now. We're able to look at structural variants in a more replicable way now, and those are some of the opportunities that we're looking at currently. So genetic drivers of late and other pathologies, we haven't really looked at in detail mixed pathologies. So the genetic risk factors of these latent HS, or latent Alzheimer's disease. We have not done that. One of the reasons that this is challenging is that for the very first picture, where we have all of these comorbid pathologies, we have to have the correct models to do that. Dr. Young presented before some nice causal mediation analyses, and we've seen some mediation analyses in other talks. Those are going to be appropriate tools to address this problem. We see shared genetic risk between pathologies. Pleiotropy is a hard problem as well, discerning between vertical pleiotropy, where a gene is correlated to a trait, and that trait then is correlated to another trait, or horizontal pleiotropy, where we have a gene that's correlated with two different traits. Disentangling those relationships is important, but it's very difficult. So that's a opportunity for us moving ahead. We saw in the last couple talks the TMEM and granulin with late and FTLD, TDP type A, and ApoE with latent Alzheimer's disease, neuropathologic change. So these models here are several of very many that exist. That vertical pleiotropy possibility, the horizontal pleiotropy, genetic heterogeneity, looking at some of the candidate genes from other neurodegenerative diseases or aging-related SNPs, looking at those in relation to late is an important opportunity that we're going to be taking coming ahead. And with that, I believe we'll say thanks. But first and foremost, thanks to the research participants that volunteer and participate in autopsy studies. It's extraordinarily important, and I guess my big gap is to encourage folks to seek autopsy consent from studies. It's a much needed gap. So thank you to the NIA and all these other people too. So thank you.
Hi, Rosa. Can you hear?
Yeah.
Do you have anything else you wanted to say preliminarily?
No.
All right. How about, I see Dr. Holman and Dr. Lee up here that were on the CUB committee. Do any of you all have anything you all want to say?
I kind of mentioned what some of the stuff before about certain genetic cases kind of looking late-like in some instances. And so there are these tweener cases, but these are highly selected cohorts. And so you just know that these exist. And there's some overlap, but in most cases, you really can't tell between late and FTLD.
Dr. Holman?
Yeah, I'll just mention, I think Dr. Rademacher showed a really nice example of when genetic data, even though the sample sizes aren't where we want them to be right now, the genetic data can act as a sanity check for the phenotyping as well. So even in the early days, when we're not up to 20,000, 30,000, 300,000 people, I think we can still think about all the biomarker conversations we had this morning and deconvolving some of that clinical heterogeneity in these GWASs, identifying genetic signals that we're confident are TDP related, and then using those as your sanity check for some of the phenotypes that we're looking at too. So I think there's opportunities with polygenic risk scores. I think we talked a lot about in the committee, those aren't ready for the clinic, but I think they are ready to be used in the way that you saw in some of the talks today, as a sanity check or a marker of some of the genetic heterogeneity that we know underlies these different pathologies.
I totally agree with that. And I think actually Michael Groth had a nice example of that with some of these imaging phenotypes that we're describing. I think looking at the genetic data can be really helpful in kind of confirming that the approach is successful or at least enriched in the population we expect it to. And I think it's an approach that, we should take more often in our samples. It's also something that could be applied in the context of clinical trial data as well, which I think would be really useful, again, in the context of some of these immune-based drugs to apply these SNPs and see how that influences outcomes.
Yeah, Dr. Jika and I fought about this with our clinical trial that's aimed towards late, about whether or not you keep people out. He's okay with keeping people out that are homozygous E4s, but for E3, E4s, he thought that they should also be considered as part of the trial, which I deferred to him. But to me, you want to enrich for people that are not going to be at risk for AD and C. So there's different sort of perspectives on that.
And maybe, can I add something about that, Dr. Nelson? Can you hear me? Yes, right?
Yeah.
Yeah, I think we talked about, like, is there already a place for a polygenic risk score for late? But because there might be some variants that have been identified that are more sort of the lysosomal component, and there might be other variants that are more, driving some other phenomena. So there might also not be a one profile or one score for late. It might be that you can tease out some of the underlying pathologies leading to the TDP-43 dysfunction, but it may be different in different people as well. So I think that going for TME and granulin might be very useful to identify 80 patients who might be sensitive to the treatments that are, for example, being now like lysosomal-based treatments that are now being generated also in the field of progranulin mutations, for example, might be very relevant. Because there's a question in the chat from Dr. Raminovici about, given the overlap between type A and late, should we consider granulin mutations, cell organoid animal models for late drug development? I think it's also the fact that the FTLD drug development, for example, that's being done might be suitable for some people or some subset of populations, especially the ones that have then the risk factors overlap with FTLD TDP type A. So I'm just trying to make the point that it might also not be a one score fits all for late, but that you might be able to tease out different parts of the underlying pathology based on different variants.
What's your experience, Dr. Rademacher, with FTLD TDP model mice and how they are good at modeling the type A disease?
Oh, yeah. Well, we don't have a type A disease. It would be progranulin. That would be the only thing. I mean, I think we are learning a lot. I think there's a lot of research now suggesting that it's the microglia that are really important. That something secreted from the microglia is inducing the TDP-43 pathology in the neurons. And of course, it's been very difficult to model microglia. So now, at least, for example, in our center, we're making these humanized models where human iPSC-derived microglia are being transplanted in the mouse brain. I think those are going to be really useful to study it. So it's really iPSC-derived human microglia that we should be studying probably. And the straight-up granulin models that have been used are difficult because, like you mentioned before, these are mostly complete loss of function models, and they actually model a lysosomal storage disorder rather than maybe FTD. So we've had that difficulty for years studying progranulin, that it's been the full knockout that was studied, and maybe not in the most relevant cell types.
We had the same, some challenges with that mouse model as well. One of those mouse models as well. Dr. Silverberg, or no questions?
We can invite questions in the room if anyone.
I can make
has
another comment that an opportunity here is if we have a well-defined clinical phenotype or endophenotype, that gives us the opportunity to do larger genetic studies. And our autopsy cohorts are probably powered for looking at specific hits there to see if they're related to TDP-43. Because, I mean, the major problem is it's hard to build up enormous autopsy cohorts, right? So the clinical criteria might be important in terms of finding the hidden heritability.
Cool. Another thing I would remark on is the ABCC9 gene is one that's very much related to vasoregulation and ischemic preconditioning. It is the ischemic preconditioning gene. And it gets to a really important point that Dr. Kawas made, is that hypertension is good for your HS, even though late is associated with arteriolosclerosis. So you think of hypertension as exacerbating arteriolosclerosis, but it raises the point there's something going on funky with these blood vessels, these old folks, that really doesn't have to do with the risk factors that we traditionally consider arteriolosclerosis one. And so, Claudia, can you comment on that?
Yeah, it's complicated, but the most important thing to say is, much to our surprise, and we're pulling this together for AAIC right now, basically hypertension, high cholesterol, and diabetes are not related to the overwhelming majority of vascular pathologies in our cohort, and that includes arterial.
So there's this period of your life in your 50s and 60s where your burgers contribute to your problem. And then there's this period of time when your 80s, 90s, 100s, not only do they not contribute to your problem, but you should be on pressors as much as possible. You should have more.
Well, I mean, this is so complicated by, if you had all those burgers when you were 50, then you probably also had 30 years of statins, and who knows what they're doing. And
then by the time you're 90, you've been dead for 20 years. Huh. Kelsey, could you comment a little bit more on this sort of complex interplay between Alzheimer's and late?
Yeah, thank you, Pete. So I think take away from the genetics of latency, for me at least, was that, okay, first, it's really, I mean, it shares a lot with FTLD type A especially, but also distinct from FTLD. It's not a FTLD subtype merely because we see a very strong association with ApoE. And second, then how is ApoE related to latency? And like we initially actually thought that it would be through AD pathology, which would be nice because through mediation modeling, we could probably show that latency has some downstream component of AD, but it was not that simple. So it's likely a pleiotropic locus affecting the proteinopathies in general, like at least A beta, tau, and to some extent alpha-synuclein, as well as TDP-43 aggregation in the context of latency. But also there seems to be some causal relationship between, at least like in my analysis, from A beta going to latency. So I think it's a bit complicated there. We'll have to dissect it. And I think the key here is to look at the actual endophenotypes, what we are measuring, instead of looking at the disease classification themselves, which may be like a more of a meta concept that combines multiple of key players. So for example, like I showed you the mediation model including amyloid beta, but with tau, we couldn't find a nice shared gene expression module. So relationship between A beta and latency may be somewhat different from between like PHF tau in AD and latency. So I think we can use genetics as a causal anchor that can guide us think more about the relationship between these complicated pathologies.
Cool. Rosa, do you have another question, or do you mind if I plow on? What do you think?
Well, I was going to say the other thing we haven't really discussed is interaction between genetic factors. And so I think the example I showed from the progranulin family is that there's this interaction between progranulin and TMEM106B, where you have to have at least one copy of the risk allele of TMEM before the progranulin mutation does something. And I believe from the work that where the ApoE data came out of that, it was also where TMEM and progranulin were studied, that also, again, the risk factor in granulin, so the 3' UTR variant, also seemed to have its strongest effect in the presence of at least a risk allele for TMEM. So we haven't even made it more complicated, but it's also going to be the combination of which variants you have together, which may actually play a role. So if you look at them individually, you might not see the same effect as if you look at them in combination.
Great. Do you mind if I ask another question? This is sort of to our folks that have wonderful imaging and have great genetics. So Dave, for example, and others who are looking at these FTLD and AD and late, how does the genetics modify your imaging phenotypes? Or have you looked at that?
We have not looked at that closely. I think again, I think there are lots of opportunities as we're discovering these risk alleles to start to bring them into our imaging models. I mean, one thing, and maybe this has been done, but with these large, older adult cognitively normal databases, I think it would be very interesting to look at like TMEM106B and how that relates to some of the sort of structural changes that we're describing, and perhaps kind of preclinical phase of structural change over time that might, help link what might be going on in the brain, in the sort of period of time prior to when people become symptomatic. Because actually, by the time people become symptomatic with late, I think generally they have pretty severe, almost like unexpectedly severe atrophy in the medial temporal lobe relative to their degree of impairment. But I think there, I know Tim and others are involved in lots of work in imaging genetics who probably can speak to this, or Liana as well.
Liana, you're on stage.
I think we have a great opportunity, but we also need the sample sizes. So I think something to consider is more. Did you call a more specific question? Maybe you should repeat that.
I think I guess the question was the imaging phenotypes and whether we're relating those to genetics.
The imaging phenotypes that relate to genetics. So I've studied, in our lab, we studied the phenotypes for Alzheimer's related genes. And with the ADNI sample size, we were able to discern what genetic variants out of the top 20 Alzheimer's genes were related to, and how they're related to amyloid, to tau, and also to atrophy in 3 separate publications. So it's doable, but again, we do need much larger cohorts, and we really don't. I mean, if you look at the SNAP cases in ADNI, how many of them can we postulate have TDP-43 pathology based on hippocampal morphology and other things? So like in an imaging phenotype, and then look at some associations. I just don't know how to go about that.
So one thing this is preliminary, but I think it gets at your question. It actually gets beyond the sample size and the case control design in that, we're actually looking at the FDG phenotype across the entire population, the whole cohort. Not just TDP and then those endophenotypes are pretty rich, and they only require a couple of variables to describe them. Actually, there's a low dimensional representation of the FDG data, and you only need maybe like 50 numbers, and you can use those as a very precise endophenotype across the entire multi-etiology neurodegenerative spectrum. And that is then you're well powered to do some genetic work, because you're not using a case control design. We're just starting this type of work. There's statistical questions to answer and things, but that's how we're approaching it. Vijay Ramanan is approaching it that way.
I think building on that too, you can use individual variants to do this, right? So it's very possible, like for example, if you look at the top granulin SNPs that you guys published a few years back in that first look at these clinical or the neuropathological phenotypes, that top variant, if you look at a soma scan, you see granulins pop up, right, in the association. So I think even though the effect sizes are small, if you've got some specificity, you can run it against like the whole clinical sample that you're talking about and pull out patterns, right, of FDG that are correlated with that SNP. So we don't have to get to the point of genome-wide significance necessarily to be able to use some of these tools, as long as you can demonstrate and replicate specificity to what you're trying to get to. So I think there's a lot of opportunity to do that. And the same is true for the biomarker conversation from this morning. The idea of some of these splicing differences that we're seeing, I can only imagine the variants that are involved in that process and modifying the TDP effect. So if you can think of it as a discovery tool to help us on the biomarker side, I think it can be powerful pretty quickly.
So
Okay, I'm getting the not very subtle Silverberg hook. But
just one more
I
question.
did see that Dr. Rademacher was going to say something.
No, I'm good.
Really?
It was me.
Here we
So
go.
how many investigators here are going to get soma scans? Because that is powerful to do it that way. And I agree with Tim. And Tim will do it on leads, and we'll do it on the ADRC samples that are, so let's combine forces and do the soma scan approach.
Cool. Well, I'd like to special shout out to our friends overseas, especially Dr. Rademacher is here. I'm seeing the background has gone from daylight to nighttime. And so thank you for your Herculean efforts here. And thank you for all to everybody. Do you have anything final you want to say,
No,
Dr. Rahman?
thanks for having me. It was, I mean, it is still a great meeting. I'll still be here till the end. Bye.
Thank you for
All
joining
right,
us.
thank you. Thank you. There's an extreme reluctance that I introduce my wing person, Dr. Jaika, who's from the University of Kentucky.
Well, that's fantastic. And what an amicable group. You know, we're supposed to be fighting amongst one another, not joking and laughing anyway. It's what I love best about our field is that everybody here loves their job. But I will say I have the best job of all because I get to take all your scientific discoveries and bring them back to patients. And for me, that's where it all comes together. So, yes, I have to hug them and cry with them, but I also get to smile and celebrate with them. And that's wonderful. So, I was certainly working with the group on presentation, thinking about this. This really represents the entire group, not myself, just myself, Ron Peterson, Risa Sperling, Phil Sheltons, Julie Schneider, who co-chaired this session as well, Leanna and Rema, who will be up next. And I think I got everybody there. So really, it's thinking about how all this comes together to think about clinical trials for what I am calling late and late plus, typically late plus AD here. So this is a major issue for the field, and I'm not going to dwell on this, but you know, all of our different groups and clinical trials was where I came in, and you know, I thought about this, you know, and it may just show my gray hair as well, that I started like Claudia. Let's step back in time to one of the early clinical trials that led to donepezil and scratch our head to say, oh my God, how did this even work, right? We didn't have a biomarker of AD. In some of the trials, we didn't even image their brain. We just, you know, do they have a memory problem? Great, we're going to put them on donepezil. And one could argue, well, that's not specific, but, you know, there was clinical criteria, as Claudia went over today, and that's a start, right? What we need to do is start to build a framework, and then we can begin to test hypotheses, but we can also begin to test medicines because we know an awful lot here, and I think we need to really move forward. So I won't dwell on those results anyway, but you know, we really have to ask that question. Not just can we develop clinical criteria or start moving late into common clinical practice, but you know, what about medicines? What are we going to do? And I think Dave alluded to this morning, you know, that now with anti-amyloid therapies, we're going to be checking CSF and doing amyloid PETs on tons of people. And what are we going to tell them when they start coming out amyloid negative? Well, I'm sorry, you probably have late, so there's nothing we can do for you. I will not be happy with that. So yes, you know, we have to think about these different things. So in AD trials, we use biomarkers to rule in the disease. We're not there yet here. But, you know, those trials are also complicated by the fact that they can't rule out late as comorbid. Current DLB trials just use a clinical phenotype. They might use some suggestive biomarkers. You may not be able to detect the TDP-43 directly, right? So we might use DAT scans or MIBG scans in Lewy body disease. And in Lewy body, most Lewy body trials, we don't even care about comorbid pathology. Give a damn. Let's just move forward and attack the Lewy body. And FTD trials, you know, frequently focusing on genetics. So I think there probably is a pathway forward on all this. So when we think about this, this is problematic. And so my only real disclosure is my little logo, my Smart HS study, safety and modulation of ABCC9. I forget what my whole algorithm is, but it's got a cute little seahorse anyway, funded by the NIH, registered and well underway. I think of that as a late clinical trial. It's looking at primary late without comorbid, ruling out comorbid Alzheimer's disease. And we do need to think about that. And we can think about, what's the population? Well, we know that 25% of non-AD autopsy cases have late pathology. So we have a sample here of people that need help. And I think that's important. And much of these other comments, for late clinical trials, really following, what's been brought up and discussed today. We also do have to think about these non-late ADRD clinical trials. We get these results that suggest that anti-amyloid therapies can only slow the disease by 27 to 40%, depending on the measure. And one scratches their head and said, well, these aren't very good medicines. Maybe they're great medicines, but a lot of our folks are running around with late still driving their pathology. And I think these are important considerations really across the board of clinical trials. So, and we've looked at this actually in our cohort, linking who has been in a clinical trial for preclinical dementia, typically preclinical AD, clinical trials for MCI or very early AD, and even our clinical trials for vascular cognitive impairment and dementia. And we find that about 25 to 30% of folks have TDP-43 pathology. They might have what we brought them into the study for. They might have AD, but this is a confound, and the same number with hippocampal sclerosis. So keeping this in mind and continuing to move forward and to think about things like, boy, what if we gave somebody lecanemab and also targeted their TDP-43? What about that? So we think about clinical trials and we think about these issues for study design. So, and I'll just use my Smart HS, Pete, my study on Smart HS as an example. So what stage of disease? What's the duration of the trial? Is it an add-on to other therapies? So in our study, we chose to go for early subjective memory complaints and or MCI with the absence of ADRD. And we thought, well, this is a slow moving disease when it's pure and we knock out that AD. So we probably should have a longer trial and treat for at least 2 years. We certainly want to enhance age. I'd love to do a study in just 90 plus year olds, but I think Claudia is going to have to do that in the future anyway. We wanted to see some evidence for hippocampal atrophy, and that can be argued, did we pick an appropriate cutoff at one standard deviation below age appropriate means? We think that's sufficient. If you're greater than that, fine, you can still get into the study, but AD negative by CSF, PET scans, and recently we discussed with the FDA, because the negative predictive value of these plasma markers is so high, we went back to the FDA and said, can we just use the plasma so we can hopefully recruit some more minorities and not give them the option of, do you want the needle in your back or do you want the radiation? Which one? Because we're coming for. Now we can just say, give us a little blood. We'll figure out what's going on. And then, of course, your outcome measures. And then you need an intervention. So we chose, on the basis of the work, that Dave Fardo and Pete Nelson did, looking at that ABCC9 gene. It codes for a potassium channel that there are medicines that can modulate and reverse those genetic deficits. So we brought in Nicorandil, not approved in the United States, but we have an IND for it, and we can already start. So we did this 4 years ago, wrote that grant 4 or 5 years ago, wrote that grant. And by golly, so if we think today, are we ready to do clinical trials late? Hell, I say we were ready 4 to 5 years ago. And we ought to move in that direction. So let's take these one by one, right? Late. We think about inclusion criteria. Well, we want an amnestic predominant syndrome. We want that hippocampal atrophy, advanced age. We want to exclude these other degenerative medical conditions. Even though we don't have biomarkers for Lewy body and or FTD, we have pretty good phenotypes to purify that sample, and that's wonderful. We start thinking about late plus studies, combo studies, people with Alzheimer's as well. It becomes a little more difficult because how can we be convinced that they absolutely have late too? Well, maybe the rate of decline, there maybe some pulling together some of these imaging biomarkers. I scratch my head on this, and even though we know that there are more people with late plus AD than primary late, I'm not sure that we're there yet, but I'd love for somebody to argue and tell me that I'm completely wrong. I do think when it comes to these studies, we think about recruitment. I wanted to put in something about diversity because this is something we encounter right now, and it's very prevalent in our community. Most people think of Tuskegee as something that happened in the 1930s, and that's true. But do you know the experiment continued until 1972? 1972 with a press release when that was stopped. So let's think about that, right? My minority participants in my local Kentucky area were part of that experiment. And those that were 60 years old were 10 years old when those media releases came out. Their parents and grandparents were potentially part of that study. So we're targeting these older folks. They have lived a long time in many, many generations and decades where discrimination and potential abuses by the medical system have been rampant. And so recruiting them is difficult. And I think this is a challenge. We're seeing this right now as a challenge when we go for our 80, 90-year-old African Americans in Kentucky. It is difficult to bring them in. So I do think that there are a lot of barriers and that we should keep these in mind. So stage of disease considerations, I think this is really, really important. And our group even discussed this. If you're going to follow these folks, well, maybe they're already at a floor very early on. Their memory and their hippocampus is already shot to heck. Is the decline too slow in late for us to even measure, right? Most of our disease modifying or disease trials rely on the fact that our placebo group is going to tank. And then that we're going to salvage that a little bit with our medication. Well, if our placebo group isn't going to tank, they're going to just slowly drift. It's going to make it much harder to do a clinical trial. And then, we can flip that coin and all of a sudden say, by golly, what about late plus? Well, that's a steam engine, that's a turbocharged dementia. So that may actually be good. Right? We can do really short trials because we know that they're going to tank over time. We do have to think with these advanced age populations as well, that there's going to be competing mortality that's quite high. I've already had people die in our study, not from the drug that we're giving them, just from natural causes. And it's not that big of a study. So we're going to have to work that in. Again, trial duration, shorter, longer. Which stage of disease are we going to really go for? And remember, the older we go, the more likely we are going to have other comorbid pathologies. So it's going to be harder and harder to do a pure late study in 80 and 90-year-olds because they're going to have vascular disease too. They're going to have some tangles. So important. I do think we need to think in today's day and age about leveraging the anti-A beta therapy for combination trials. And you know, I think we should talk about how we can do that, you know. So if these drugs get full FDA approval and Medicare coverage, there's going to be a lot of people that want to be on these medicines. We see them in clinic all the time. Is there a possibility to look at these folks and say, hey, wait a minute, even though you're on the medicine, you're still spiraling out of control, moving way too fast in your decline. There must be something else here. Maybe we can apply some of these other late algorithms for a probability that they don't just have AD, but they have late as well, and add and build a combination clinical trial. I really think it's a lost opportunity if we don't think about this, and now's the time really to plan for this sort of thing. Oops, my clicker stopped working. Okay, let's see what I got here. All right, very good. So also let's think about leveraging screen fails from ADRD trials, right? This is the same thing that we're going to see with clinical implementation of anti-A beta therapies. People come in, and this is just our average screen fail rates at the University of Kentucky. We're bringing people in for a preclinical Alzheimer's disease trial. 90% of them are failing. They're not amyloid positive. I don't know why. Given how many people are amyloid positive out there in the world. Our MCI trials, 60% of people are screen failing. They got something else causing their MCI. Our dementia trial, still 30%, you know, we turn people away. And I have this discussion with patients all the time. Well, then what do I have? If you're telling me this isn't Alzheimer's, what are we going to do about it? So we think about outcome measures as well. Now, we chose for SMART-HS, we chose to stick with memory and MoCA, a general and a memory measure. We're hoping that we're getting these folks early enough, but it certainly is a confound, you know. If they're already at floor, we're not going to see any change over the course even of a 2-year study because they're already at the bottom of the barrel. Hippocampal atrophy is a primary outcome measure for us. If their hippocampus is already completely at floor, we're not going to see any change. And if we're not going to see any change, we're not going to see any benefit. So we do need to scratch our heads. And I think this comes back to something that was discussed this morning in terms of clinical phenotype. Remember, for no degenerative disease is there one clinical phenotype. And that's not because the disease is heterogeneous. In one person, there is no one phenotype. The phenotype changes every year. Every new time point, their phenotype changes. So we really need to keep that in mind, thinking about outcome measures. You know, for later stage or more advanced late or late plus, we may need to think about adding some of these other measures, language, some of these other things that we see not at floor, but still moving during that period of time that the patient's engaged in the study, and other novel outcomes. So we've actually seen many of these figures today presented by some of you all, but lots of different things, thinking about new neuropsych measures, MRI measures, other imaging modalities, FDG, TDP-43 PET, if that's ever developed, and fluid biomarkers. So there may well be many other paths forward. And I think really that this, hopefully it will stimulate these biomarkers to continue to be moved forward. And then, you know, we think, you know, well, what if we have, maybe it was a fluke that, you know, Pete and Dave identified ABCC9 and we found that it was druggable. Maybe there are no other targets for intervention. And all I can think of, and, you know, David raised this point today as well, what about cholinesterase inhibitors? What do we tell people? Do we know if they work? You know, well, this is just a screen of cholinergic deficits in different diseases. And you guys can see up at the top, HS aging, HS with other diseases in the red when it comes to choline acetyltransferase. So it should respond. Wouldn't it be nice so that we could answer our patients, yes, you don't have AD, but you're still going to benefit from donepezil, like we use it in Lewy body disease and other disorders. I think we need to think age is a major risk factor. What about some of the senolytics that have been developed and are already out there in clinical trials? We're using nicorandil, targeting this potassium channel. What do we got out there that could target TMEM106B? Or granulin, proteinopathy based, right? There are medicines out there right now, anti-TDP immunotherapies, verdiperstat, tamoxifen, back to the estrogen story, although I think that would go against, Claudia, your data. But there's some, at least in vitro evidence that tamoxifen might be helpful in reducing TDP43 pathology or T3. So I don't have an acknowledgement slide, but I would like to acknowledge all of the other team members. As always, my colleagues at the University of Kentucky, Pete Nelson, who dragged me kicking and screaming into the world of late. And but I'm just so delighted to watch the growth in the field and really encourage everybody out there. Let's pull our science together. Let's get some of these medicines out there. Let's start helping these people, not just talking about them. I think that at the end of the day, that's what we all want. So thank you all.
So the next speaker is Raman. I don't know how to say her last name. There you are. Okay, thank you.
Okay, I get the pleasure of being the last speaker today. I'm going to take a slightly more general view of this entire topic of clinical trials. The full day we've been talking about specifics, but the different aspects of clinical trials are things that need to be put in place before one can even think about designing and then conducting a trial. So the committee, the part of the committee that what we focused on in this particular area was really how do we actually design trials? More importantly, how do you conduct it? How do you actually successfully conduct a clinical trial in this population so that at the end of the day, you have a successful trial and hopefully a successful drug. But even if it's an unsuccessful drug, you still have a successful trial. So that's sort of the way in which we've approached this. And again, as Greg mentioned, this is part of the committee. I'm representing the committee on this topic. And the way we sort of laid it out is around 4 essential design considerations. Around recruitment and retention, we've talked a little bit about it, but I want to get into depth around what it means to recruit and retain participants in this particular subpopulation in a trial. And then talk about some of the design elements, statistical considerations, and operationalizing everything that all of you have mentioned today is really going to be very, very critical to conduct successful trials. I do want to spend a little bit of time talking about trial operations and then hopefully bring this all together with a summary of where the subcommittee felt we were at this point. So without successful recruitment and representation, we aren't going to be successful in finding treatments that are generalizable. And so the recruitment strategy, informed consent, and eligibility assessments, all of which drive successful retention and recruitment strategies. So I wanted to share some data that I was looking up. This is new to me. So I was trying to figure out what this population is, who these individuals are, and I was looking up some information around older participants in clinical trials research. And the first paper I found was the paper on the prevalence of individuals by age. So this chart on the left is looking at less than 65, 65 to 74, 75 to 84, and 85 plus. And the dark bar reflects the clinical trial participation in these age groups, and the light-colored bar represents the estimated number of people with AD in these groups. And it's obvious from this chart that as you get past 85, the representation in trials is really, really low. And as we've heard throughout the day today, a big part of the late population are individuals who are likely over 85. And part of this is because our inclusion-exclusion criteria prevent individuals over 80 to even participate in our trials. So question is, do we have the infrastructure in place to be able to conduct trials in populations 80 and over? The second table is from another paper by Banzi et al., by Shenoy et al., and they talk about some of the reasons for underrepresentation of elderly participants and patients in trials. And I found this very interesting because I think this is really important to think about as we talk about what do we need to conduct trials in late. Talking about challenges in gaining informed consent, the multiple comorbid conditions that may pose challenges in outcomes, polypharmacy, some of you talked about it today with drug-drug interactions, challenges in compliance with study procedures, which become very important in analysis, age-relevant formulations and packaging as we think about the actual interventions that we include in our trials, fear of failure due to confounding behavior of drug in elderly populations. And now as you think about if you have populations in their 60s, 70s, 80s, and 90s, these confounds could play a big role in effects. Sponsors fearing higher cost of medical management and compensation in this population. Institutional and logistic problems. Supportive care may be needed, and then investigator preferences and perceived difficulties in screening. And as I just mentioned, all of our trials right now have protocol restrictions with exclusion criteria on age. So we don't even know how to work with clinical trials in this population, at least based on my experience. The second challenge that we've had, Greg already alluded to this, and that is the fact that we haven't done well in having representative samples in our studies. So our results as we now developing successful drugs is that we still have to ask the question about external validity or generalizability. And so the upper left is really data from some of our preclinical and early AD trials that show that there is a disproportionate screen fail rate among individuals who are Black, Hispanic, or Asians compared to whites in our preclinical and early AD trials because of levels of amyloid in their brain. So you've already got this disproportionate screen fail rate. And on the right is some data from some meta-analysis that was done both in disease-modifying AD trials as well as dementia prevention trials, which show you the proportion of participants who report as being Blacks, Asian, or Hispanic in our trials. And you can see how small those numbers are. The large numbers are really because these trials were predominantly done in parts of the world where these populations were the majority population. So we have a lot of work to do in terms of generalizability of our sample as well. I'm not going to talk too much about study design because Dr. Jaika did a fabulous job of addressing all of this, but these are all elements that need to be taken into account. They have to be operationalized. It's one thing to say that we would like to do this. It's another thing to say, how do we do it in the context of a trial, a randomized multi-center trial, not just in specialized institutions, but in institutions all across the country. The next part, which is near and dear to me, is the biostatistical considerations. All of this is great, but okay, now what is the sample size you're going to need, and how do you evaluate effect sizes? How do you know that a drug actually works or a treatment actually works? And so this is something we've been dealing with in terms of even our AD trials right now is a randomization strategy. As we now have these anti-amyloid treatments coming into use, clinical care, is needing to stratify on use of anti-amyloid treatments. But then we also have to stratify on so many other things. And so the complexity of randomization increases in our trials. And now we've got age to worry about as well in late trials. So the randomization strategy becomes a lot more complicated as we start thinking about these trials. Biggest challenge, I still think at this point, even after listening to all of you here today, is sample size and power. And that is, how do we find the most effective sample size to be able to answer our research question and what is that effect size that would make the most sense. And the reason this is challenging right now is we don't appear to have reliable estimates of treatment effect. Forget about treatment effect, even effect in a control population right now. And the variability of the potential outcomes. One, what are these outcomes? Biomarker, clinical. What is the variability of these outcomes? And what is this variability across the different age groups? Second, and this has been alluded to during the day, which is heterogeneity of the disease and risk factors in the population. And this heterogeneity is really going to play a big part in determining the sample size and the effective size for our trials. Heterogeneity of disease progression, I'll show you the next slide, which shows some of the data around the challenges that all of us looking in AD research have regarding the heterogeneity of the progression among our patients and participants. And then we do have to account for this Hawthorne effect, placebo effect, as you get older participants into the trial. And all of this means the analysis strategy is not easy. And as a trialist, my ideal analysis would be a 2-sample t-test or a chi-square test. Just in an easiest sense, your analysis strategy should be the easiest and the most simple if everything else works according to plan. But our analysis is not going to be that straightforward here. It's not just longitudinal modeling, but you're now going to have to account for all of these stratification variables, all of this variability, background medication, and these potential differences in long-term trajectories. So there's a lot of work that needs to be done, I believe, in this area still. This is a busy slide, but it's got a very simple point, and this is work from Dr. Shelton's group, and it really looks at the group differences. So I'm just going to take a minute to just orient you to the slide, but let me just stick to the top left, the A's. The first 2 plots are simply along longitudinal trajectories among those who are 65 and older and those who are 65 and younger, less than 65. And as you can see, there's variability in those trajectories. The plots below, the histograms, are really the group differences. So it's a simulated study. They've done about 10,000 simulations and looked and created these randomized trials. And they've looked at these group differences in CDR sum of boxes to decide whether this treatment would have been effective or not. And you can see in the greater than 65 how tight that effect size is. And then you get to the less than 65 years, that range becomes much wider. So you have this broad range of observed group differences. And what this translates to into in a trial is what is the sample size that you're going to need to be able to detect this heterogeneity of group differences with sufficient amount of power and confidence. And so this data really reflects the heterogeneity of the longitudinal change and the effect sizes due to treatments that we are likely to have and we have in our trials. This is some work that we did with some of the AD trials, and this has direct relevance to age that is important for late trials. And on the left are the trajectories of change in the ADAS-Cog over time, and the sample sizes, and on the right are the sample sizes needed to detect these changes. And we looked at 4 age groups, 51 to 71, 72 to 77, 78 to 81, and 82 to 94. And I think this is the data that is very relevant to the late population. And you can see those different trajectories in these different age groups. So depending on who you include in your sample, in your study, you're going to have to account for pretty different trajectories of change depending on where they are in their age spectrum. And on the right, the plots tell you the increases in sample size that are going to be needed as you have older participants in your sample. And this is because there is much smaller, the rate of change is much lower, and therefore you're going to need much larger sample sizes to detect the same effect. So something we have to keep in mind as we design our trials. From a coordinating center perspective, the success of a trial works only as good as the operations of a trial. You can design the best trial, you can write the best protocol, but if you cannot execute that trial, you're going to fail in answering your primary question. So I do want to take a couple of minutes to talk about what we as a group thought about in terms of trial conduct. The first is capturing relevant data. We're dealing with individuals who are older, which means you want to capture only relevant data and nothing more than what is needed. So data management becomes an important element. Moving closer and closer to pragmatic trials allows the burden to move away from the participant and makes it easier to conduct these trials. I don't know, can we do pragmatic trials? Can we move towards pragmatic trials? Might be difficult if we are going to be asking people to do scans and all of these cognitive measures and all of this other data among people who are older in age. The study composition, I'll talk about it in the next slide, in terms of the investigative team, has to be expanded to include other disciplines that we don't traditionally have. We actually have HIPAA issues for individuals over 90 years. There are regulatory issues that we have to think about as we do trials with individuals over 90 years of age, and the safety review process. So what must be considered in trial operations? Because you're dealing with individuals who are older, there are other elements of data that we believe needs to be collected in addition to our traditional data. Things like function, comorbidity, social supports, which may all play a big role in how individuals respond to treatment. Collection of forms have to be simple and easy to use, can't be complicated. Execution of trial operations has to be very, very smooth because this is a population that is not going to be comfortable with complex procedures. From a statistical perspective, this is what really worries me, is missing data. Missing values due to difficulty answering questions, refusal to take exams, being tired after long assessments. So all of this could result in missing data that could be informative, and knowing the type of informative data missingness is going to be critical for the analytics of procedures being used. Expertise in geriatric medicine becomes critical in the design and operation teams in a way that it may not have been before. This is the HIPAA safe harbor rule that I think we really do need to recognize and understand, and that says all ages over 89 and all elements of dates indicative of such age, including birth date, except that such ages and elements may be aggregated into a single category of age 90 and over. So if we are talking about populations that are older, you can't just clump everybody who is over 90 into this one bucket. And it goes completely will violate HIPAA if we do. So as a field, we are going to have to work towards addressing that particular problem. And then you've got safety issues, pharmacokinetics, pharmacodynamics that might potentially differ compared to younger participants. So what does all of this mean in trial designs? So I'm going to just wrap up from our perspective of where do we stand right now. I think I'm as optimistic as Greg was when he ended, which says we do have the infrastructure in place, and this group and the group online is really testament to that fact. We're here, we're thinking about these issues, we've got the right people at the table. We do have to leverage our biomarkers that are used for screening in existing AD trials, identifying those amyloid negative screen fails, maybe getting that MRI scan to establish hippocampal atrophy, and then being able to find these individuals for late trials. Using p-tau positivity to guide location in the AD spectrum depending on the level of cognitive impairment. Representative samples and trials continue to be critical, but we believe that the biggest challenge to the work that we have to do is the heterogeneity in Alzheimer's disease. It impacts the sample size, we are heterogeneous in terms of the impact on age, our inclusion-exclusion criteria. We have very little data on race and ethnicity. We don't know what the trajectories of change are in different populations, and our outcomes are still very much a work in progress. And so at the end of the day, we believe at this point, there is a need for a well-characterized observational study. I personally will take it one step further and say a trial-ready cohort where you really are characterizing these individuals to be eligible for trials to guide appropriate study design, power, and sample sizes. And so again, with that, just want to talk about the behalf of the subcommittee. This has been one of the most fun things I've done in a really long time. So, so thank you.
Rama, can you stay up there? And Greg, can you join Rama?
And I know Liana and I see Risa from our subcommittee.
And also Ron, I don't think he's here anymore, but he was on the committee, and the other person we should call out is Phil, I don't know.
He's not here either. I know.
He's not here,
no.
but I didn't know if he was online.
Is he online? I hope he was.
Not online?
Yeah,
Okay.
and then of course Julie.
Oh, of course, yeah.
Yes.
So I'll start off by asking about something people have been bringing up throughout the day is the screen failures for the trials. Is that something that could be potentially utilized?
I think, I will just comment about our real world experience. We are utilizing that right now for enrollment into Smart HS, and vice versa, actually. I don't know why it is, but we recruit somebody for Smart HS. They always are amyloid positive. We recruit them for an Alzheimer trial. They're never amyloid positive. So we're just flipping them, once they come in. And it actually works, and people are actually quite pleased because they've gone through their disclosure. They have a clearer idea. I tell them we don't yet have a definitive biomarker for late, but this, given that we're not seeing AD positivity, this is the way that we may be best able to help you.
So you're doing that, but what's happening in the broader
I think-
community of people? Do those people just go away and you never see them again? Or are they being followed?
Risa. She's
Yeah, I was gonna, so, first of all, I think it's a fantastic idea. So I really support what you've talked about, Greg. We are, for ahead following them, but they are asymptomatic individuals, so they may be tougher, but we can think about whether there are ways. But for symptomatic trials, I think we should find a better mechanism of capturing these people nationally and offering them at this kind of a study. And the other thing is the Alzheimer's Association for ALZNet. They are now doing this in the clinic and are going to be capturing a lot of data. And there may be ways, which will be many more people than screened for trials, potentially millions, if there's reimbursement anyway, that there'll be millions who are negative, as you just said, Greg. And so finding something to offer them would be awesome. And then go ahead, Julie, or bring this up, but I wanted to come back to the combination and addition, because I think also thinking about going after the people who have a little bit of AD pathology, but TDP-43 is driving their some of their decline is something we should talk about. Maybe that's step B or it's step F, because that's going to be later. But I think there's opportunities for that as we build these platforms for amyloid plus tau, as you mentioned, Greg, but why not amyloid plus TDP-43 or amyloid plus tau plus TDP-43?
So let me ask a question, Risa. So right now in the treatment realm for Alzheimer's disease, what happens when you see somebody who's amyloid positive, but you think their T is not high enough to really be driving their cognitive impairment?
Yeah, so again, I think the Lilly donanemab results is one of the first times this has come up, because as you saw those results, they published in the intermediate group because they screened on tau, and then they also looked at, or not published, presented or mentioned the high group, but they didn't talk a lot about the group they screen failed on tau. And that would be an incredible group to go after for late, because it's initially in phase 2, I know it was about a third of participants who had amyloid but didn't make their minimal tau criteria. I don't know their phase 3, but that's a really good question to ask. And those people were screened, some of them not that long ago. But clinically, we don't usually get tau PET to decide about treatment yet, but donanemab could change that. We'll see.
Just to follow up on that, because I think it's a really good point. And, the difference between the donanemab and lecanemab trial was, the requirement of tau PET. And I guess, maybe anyone can answer this, but, are we at a stage now, given everything we've talked about today, that if we're doing a symptomatic trial, that's directed towards AD pathophysiology, we should only include if tau PET is positive. Particularly if the syndrome is amnestic and there is concomitant, structural change in the medial temporal lobes. Those individuals are, more likely to have TDP or some other driver of their symptoms. Now, again, we can argue whether amyloid clearance might be beneficial to them as well. But I certainly think we need to think about stratifying or identifying or maybe excluding or doing separate trials in those individuals versus those that are tau positive.
It would seem, and I'm not a trialist, but it would seem to me just that you should power and do both. So I don't know if that's been thought about, but why not do both?
Yeah, so I think this is something that we are going to have to do moving forward. Right now, until now, we never had data to be able to do this. We didn't have trials that were completed. Now we have trials that are completed. Once we get out, and this is where data sharing comes in. Once these data are now available, either as part of the study team or not, you can start looking at these questions. Is the change in certain groups different from others? Across certain subgroups different? And then what does it mean for the power of a future trial, assuming the distribution, a certain distribution of these things? Which gets me to the other point I was also wanted to make, and that is the power of data standardization across all of the, say, NIA-funded programs, so that it doesn't matter whether you are a screen fail from a trial, whether you're being seen in an ADRC cohort, whether you're an ADNI participant. If the data is standardized, you can bring all of these data together and effectively ask and try and design our trials better. Right now, we still do things very differently. We are trying to, but it's still a work in progress. And so as these new biomarkers are being developed, as all of this innovation is happening, it would be great to start thinking about standardization so that you're sort of not repeating these learnings every time.
So Rima, I have a question I'd like to ask. We've heard today that late is relatively indolent compared to Alzheimer's disease, possibly more indolent than late Alzheimer's disease, which would mean therefore that your slope is very small. Mm-hmm. The attrition rates are going to be high, mm-hmm. likely because of the age mm-hmm. and comorbidity. But in fact, and I think your simulations were based on late life AD.
Right, that's correct.
Whereas there are cohorts, for example, Claudia's cohort, where there's probably data on people who were characterized as AD in life, shown to be late subsequently, mm-hmm. that one could go back and use that data to look at, to generate essentially expected rates of decline for individual neuropsychological measures. Yep. That can then be used to plan trials.
Absolutely. I think that's the only way to do it, is find cohorts that are already in place, identify individuals who are late compared to those who are not, and actually look at what these trajectories of change are, and then use the appropriate trajectory of change to then design a trial.
So I think there's a bit of that out there, and some of that was shown from ROSMAP today, really derived from the autopsy cohorts. To combine that, but I did want to get back and say, this concept of who cares if they're A+ or not? It's about the driver of the cognition. I think is really, really relevant. And one of if the tau platform for ACTC is ever funded, we may have combination therapy there targeting both amyloid as well as tau, and we will have the measures to know who's tau positive, who's at a high, low, or medium range, and it may actually help us. But when doing that trial, we should be keeping late in the back of our minds and try to learn lessons that can help us move forward.
I just wanted to make one point about the A positivity there. I think there's several ways in which we've been talking about pragmatic trials, using LSNet, expanding. And a lot of the thinking about how we're stratifying and telling people about trials begins with the question, A neg, A pos. I think that's a mistake. And I see us continue to push that way. But just if we actually phenotype them differently, functionally, anatomically, syndromically, Then when you ask questions about proteins later on, you can bin them more properly. If you think someone's a limbic predominant condition, then you don't even really need to ask the A question. You really are asking the T question. And we've learned this actually from Leeds as well. Many people, they get enrolled into that study. They are presumed to have Alzheimer's disease, but we find a good number of them are A and T negative. And so do we presume they have late? We do not. I mean, they're young, and we actually don't know what they have. And so when we're looking, we're still working to figure out who all these people are, who we ask the protein question immediately, and the protein question's negative, and then we don't know. So as we think about these larger trials, ELSNet and screening in our clinics and running it like oncology, we need to do a little bit more precision phenotyping up front about the clinical phenotype, rather than jumping right to cognitive problem, what's your A? I think that's a big problem that I see the field heading that way. And I think there's several reasons we discussed here why that's could be problematic.
It's all Cliff Jack's fault for ATN. It should have been TAN, right? Is that
Well, I also think you have to think about the treatment, right? So a lot of this has stemmed from this being anti-amyloid treatments, right? But there's a lot of treatments out there that are agnostic to amyloid or tau. And I don't know how they enroll in those, but those certainly should have a bigger bucket of people.
Well, and in clinical practice right now, there's lots of treatment, management, counseling that goes on that does not revolve around amyloid lowering. And we need to continue to do that. We actually need to get better at that, including ventricular peritoneal shunts for neuropressure hydrocephalus, which are an underutilized treatment strategy. And reducing, and as we expand these thoughts to clinical practice, rather than just the research realm, which that's what we're talking about with pragmatic clinical trials and with ELSNet, we can no longer use this first question, A, negative or positive.
Yeah, and I think that's, for me, where the trial-ready cohort has to come in. You have to be able to characterize people, and we don't know much about characterization by genetics or race or ethnicity, whether you want to use the social constructs or not. But we don't have much information about all of them. So we're talking about diversity and making our sample much more diverse, but we're not powering our studies taking that diversity into account. So I think you've got to address it both ways. You've got to know what the changes in representative populations through our cohort studies, and then use that to really effectively power our actual trials. But you had a comment?
Yeah, I have a question. I'm so far from trials, but what's better? So I agree that the problem in screening for A- is that's assuming that the biology is the same for pure late versus AD late. We don't know that, but in terms of being pragmatic, is it better to be an A negative, highly enriched late cohort that barely progresses, or a mixed cohort with Alzheimer's, with late that progresses very fast, right, but is more heterogeneous? I just don't know from a clinical trial standpoint which one is better.
And some of the data from that paper, what I saw was the decliners. It depends on the proportion of decliners that are driving some of the effects that you're seeing. And so the larger the proportion of decliners in your trial, more possible that you're going to see that treatment effect. And if you don't have as many, it's going to be much harder. But again, I think it's now that we're getting these data, and so I think being able to look at these is going to be important. But I know Gregor.
Yeah, I think I think we're not going to. I was trying to really make that point with my slides where I had late and then late plus. These really are two different populations. I don't think we're going to be able to do trials for late unless they're purely based on biomarker outcomes, because when we have late plus AD, their slope is worse than even AD. And when they have late alone, it's much, much, much slower. If we mix those together, the degree of variability and heterogeneity
that's where combination therapy is something to think about,
yes,
right?
Yes, potentially.
So I was going to also bring up, in your trial, you wanted to exclude all E4s, a quarter of the population.
Oh, no.
I wanted to.
I'm looking at you. I'm looking at you.
I did not want to because
Yeah, so you wanted to. So also, we know E4 from hide and seek and from others is very much related to TDP-43 in late. Maybe they're the ones who are declining. And now you're taking them out of your study, or you wanted to. We have to be really careful about the exclusions that we're making and the presumptions that we're making when we start excluding people.
I think we see a large number of folks that are ApoE heterozygotes that are amyloid negative, and though there's something else going on. And as was mentioned earlier, ApoE is a risk factor for just about every disease of aging that we know of. So it could be driving the late, just not AD for other unexplained genetic or environmental exposure risk factor causes that we don't yet know. That's why I argued with Pete and said no.
But this is the rubber meeting the road. This is why we're here.
Exactly.
And if we can make recommendations and contributions and come to consensus about what is important, what are the parameters that are important, what are okay to, not okay to exclude on, that would be a very valuable contribution going forward. It may change over time, but that's okay. But we need to have some guidance.
Yeah.
Risa has her hand up.
Oh, sorry. I was just totally agreeing on the ApoE, so I'm glad you did not exclude them. And similarly, in the future, really thinking about whether you would allow people in who had below a certain level of amyloid, but maybe were not, quote, amyloid negative. So if you're in MCI or you're dementia and you're below 40 or 50 centiloids of amyloid, I have to question whether amyloid is the only driver of your dementia. And they are potentially a great group to test. I agree with you about the heterogeneity. I wouldn't necessarily mix those with pure late. But in terms of a future design where you want to capture, because there's a lot of older people, Claudia can speak to this, but who have a little bit of amyloid, and the neuropathologists, they have a little bit, and they might be the very group who would respond to a therapeutic that would go after TDP-43, or again, to push this idea of something more ubiquitously going after protein aggregation.
So how about a platform trial? Let me complicate it further.
Yes,
Where you
yes.
have your pure late, and then you have your amyloid positive late, and you have an amyloid plus TDP treatment and only TDP treatment, then you need a placebo. I mean, it's obvious that you might need that.
I think probably in the future that maybe where the field goes. I think right now just getting started is what's important. Getting started with clinical diagnosis and bringing late into clinical practice, getting started with clinical trials. That's all we need. And then it's going to carry itself forward.
I think we have time for one more question online.
We got a question, and maybe you understand this, I didn't fully. Is there a hope for targeting MDT against TDP-43 and target MDT against tau? We asked for clarification, but haven't gotten a response yet.
Tim?
No.
Maybe it was supposed to be DMT, disease modifying therapy.
Am I?
Actually,
Yeah, that's a good question.
It could be. I think we talked about that a little bit, and I do think there's possibilities for disease modifying therapy as I went through. I put together that short list of possible trials we could do in the field of late. It took me 5 minutes of thinking. That's several R01s I could get out of that, I think. Some disease modifying, directly affecting TDP43, others influencing, like our Nicorandel study, influencing these potassium channels that somehow interact with the vasculature to contribute to hippocampal sclerosis, as Pete showed in his genetic talk. That's disease modifying. It's reversing, in effect, not treating a symptom per se. But I do think we need to do the symptomatic studies as well. I think, there is no harm in doing a small study just to say we have evidence that the nucleus basalis is affected with TDP43. Let's just do a little trial so that we don't have clinicians out there in the world not taking care of these people. Turning them around, sending them out the door, and doing nothing for them is a real problem. I don't like it, and it creeps in. I want to fix it.
Okay, I think that's all the time we have devoted to clinical trials subcommittee, but we have a lot more time for general discussion. And I think this is really pulling everything together, and basically saying at a start, what Dave proposed and what we agree with is that we're ready to make a clinical diagnosis of late. And can we pull things together at this point, clinical biomarkers, with or without genetics, to allow that to happen and allow there to be clinical trials. So, so I want to just open it up for discussion at this point on what everybody thinks about that.
I just might add, and it would be great to get people's thoughts. I mean, I think it really is important to think contextually about late as the disease driving, pathology, and when late is more intermixed with Alzheimer's disease or other conditions. And the degree to which we feel like in those different scenarios, we have confidence in a clinical diagnosis using the tools that we've talked about today, knowing that, again, in the context of AD, our certainty is obviously going to be lower than in the context of, where someone without AD or without another clear neurodegenerative condition. But I do think making that distinction and thinking about the degree to which we have confidence in that and our readiness for that also has real implications for clinical trials like we just discussed, and pragmatic studies like, ALZNet. And I guess one other thing I'll just sort of say, and I think there's obviously practicalities and issues around this. I do think from everything we talked about here, we often say, well, in practice, we don't have all these tools. I think now that we are doing disease modifying medicines, we all need to kind of advocate for using these tools more frequently within our practices. And maybe that, is, policy related and otherwise, but I think we need to start to apply some of the techniques and modalities that really, actually enhance our ability in our research cohorts to make better predictions about what people's underlying pathology is.
So
and maybe we could change the angle of the camera, because this is not my best angle. Thank you very much.
So you look great. You look great. No, so I would throw out also this idea of research criteria versus clinical criteria, because right now many of these, other than structural MRI, most clinicians are not going to have access to any of this other stuff, right? They're not going to be able to do it. Medicare is not going to pay for it. So what do people think about that distinction on research diagnosis, clinical diagnosis? Right now, no one's, other than Dave are making the diagnosis, of late. or very few. And I'm sorry, and Greg, and there's a handful of people who are making the diagnosis. But do we want to restrict it to the people in our research? I mean, we can have separate sets of criteria. If you're doing research, this would be ideal. And if you're not doing research, this is what you can do to get a probabilistic criteria.
Let me, I'll answer one. I think this will ease some people's minds. If you, like my criteria said late CS, right? Like late clinical syndrome. Let's change the name to, limbic, predominant degeneration syndrome. Now that doesn't have a protein associated with it, but everything else still applies, right? And then you can think of it like posterior cortical atrophy. I don't like that name, but once again, that's a syndrome. It doesn't have a protein associated with it. Is it Alzheimer's disease? 100% of the time, it is not. 80% of the time, it is. So we usually have those clinical criteria for posterior cortical atrophy. And then if you want to say the etiology is Alzheimer's disease, you get a biomarker. But so I'm just saying that there's precedent. And so you can, and then the other question would be, can this be done routinely? It's like, that's why in the criteria, the core criteria is amnestic dementia syndrome, and then 4 routine points is just, which only require routine information from a routine evaluation, history, detailed patient history, physical, a brain MRI, and some sort of cognitive testing, which can be done at the bedside. Just using that portion of the criteria, you can get to moderate likelihood of having the syndrome associated with TDP-43, moderate likelihood. That's pretty good. If you want to get to high likelihood, you will need to do one of 2 things based on that criteria. Prove tau negativity in some way. That's not hard to do by current and emerging things available to people. So that can be done in routine practice. So I just outlined a way to get to high probability, late CS in routine practice. You don't have to use any fancy techniques to get there. The only one that maybe you may need a fancy technique for is FDG. Why do you need a fancy technique there? Because people can't look at the scans and interpret them. Why can't they do that? Because they're not doing that. If they did that, they get good at it. It's not hard, but you would have to train yourself to do it. Or we can put analytics and quantitation on top of it. That would require some advanced analytics. So, and I think as a field, we should do that for our patients. I don't know why we wouldn't. So I don't think we should shy away from being more precise about what's causing degeneration of the mental functions in our patients' brains. We should do that. So the criteria, as I outline them, and I have taught them to geriatric fellows who rotate. They're not interested in advanced testing. They are using them. They can use them, and they don't need that advanced testing.
Dave Jones, question: the same person that came to a conclusion could be moderate likelihood of TDP-43. Wouldn't that person also be moderate likelihood of Alzheimer's?
That's why it's moderate.
Yes.
And then that's why I said if you really want to get serious and go to high, you need a T-neg. Then you cut all those people out.
P-tau 217.
No, see, p-tau if it's negative.
Yeah, exactly. If it's negative
is good, but then if p-tau is positive, you still don't know.
If it's negative, it works for late. Yeah.
But so to Liana's point, so if we've got both these groups that are moderate, before we add in another clinical syndrome name, at the end of the day, they have a progressive amnestic syndrome. That fits, it works. But then what we're trying to predict during life is what their likely underlying neuropathology is. And so then you get to your moderate likelihood through the tools that you're using, but you've kept it simple. So it's more public facing and that you've got a progressive amnestic syndrome.
You're in between. So you can't say one more likely than the other unless you have a blood test. And I think we'll be equipped with blood tests maybe next year.
I agree with everybody here. And what you're really talking about now is nomenclature, which we've had a big committee discussing. And we agree. I mean, the syndromes should be separated from the pathology, but that's not real world practice right now. That would make a huge change. So having said that, I'm going to move to Cindy Carlson, who has a question online.
Hi, everyone. Sorry I'm not able to be there in person. I've been kind of intermittent today, but yeah, I think this ties in with a lot of the things you talked about the ADRC meeting too, though, is just thinking about how can we move the ADC program so that we have more representation and that we link it better to clinical trials. And if we are getting, making sure that our Alzheimer's centers don't have upper age limits for engagement so that we are recruiting older adults into here, that we're following them longitudinally. And then with the UDS4, we are trying to move toward what we've been talking about the, not necessarily separating out, but describing the pathology and the biomarkers and the clinical syndrome better and capturing some of the, behavioral changes and some of the other cognitive, motor, other clinical syndromes. So it seems like it's a good point to bring these patients in to improve their representation, increase the ages, follow them longitudinally, capture the messiness and the comorbid illnesses. And that's where we're going to get our, and then this whole idea about instead of recruiting them into a late study, and like Greg was saying, you either recruit them into a late study and they have AD, or recruit them into AD study and they have late, instead to have where you let them evolve in their pathology and their clinical syndrome, and then you screen them and see which trial they're eligible for. So it seems like there's a lot of good infrastructure and tying together things that have been talked about at the ADRC meeting with what could help with clinical trials with late.
I think that's a great point, but since we have you online now, Cindy, because you're part of NAPA, and I think one of the things, we're all recognizing that it's about the clinical syndrome, and that's been brought up numerous times, but AD is ingrained in our society. NAPA is all about Alzheimer's disease. Right. And people think of that as the amnestic disorder of aging. Do you have any thoughts that you want to share with us, having, chaired that council?
As far as how to change people's thinking about it, or which is hard?
Well, I'd just like to hear your opinion on, it's difficult to change names, right?
Yes.
Very difficult. And do we try to do that, or do we just move forward and say, as neurologists, as experts, do we just move forward and just we gave TDP a new name now so that it had a name. So we have late, we have Alzheimer's disease, we have ATN. Do we just move forward at this point and say it's late, or it's, AD by ATN, rather than trying to change the nomenclature of a limbic disease or an amnestic syndrome? I'm just, from a practicality standpoint, asking that.
I'm fine with leaving late. I mean, I just suggested that because there's some intellectual hangups for having a clinical syndrome defined that way, especially probabilistically is still not necessarily going to be TDP-43 every time, but as we showed, it'll be enriched for that. So if to reach consensus, if it takes changing a name, that's something I'd be willing to do.
I think it's hard because we've talked about this a lot now, and I see Angela there and Ron Peterson and others. So, hello. And I know the nomenclature group has done so much work, and just in all of medicine, we're trying to move more towards descriptive nomenclature so that we're not using, even the term Alzheimer's or Parkinson's disease or things like that are, tied to people's names. So, but we've talked a lot about this, and scientifically, it makes sense to describe it using, definitions like where it's occurring and what the pathology is, just like, and what a clinical syndrome is, so separating that out. But then, and I think it's really up to the scientific community then to make it something that the clinical community can use, so that it can be, it's not a separate type of clinical syndrome or separate nomenclature, but something that's simple enough that clinicians can use. But then adopting it, I realize there's all kinds of politics around adopting the nomenclature. And then, as our federal partners know, the nomenclature has to get into the legislation to support the funding. So if there's funding for late, that has to be, or if there's funding for Alzheimer's, it has to be under that. So in order to get that new terminology into the legislation, that takes another process. So I think it's like this process of starting with the scientific group, like meetings like this, trying to describe the pathology, how it links to clinical syndrome, and then moving it into helping the clinicians to understand the big picture concepts, not all the details, but big picture concepts, and then moving it into, getting into legislation and the advocacy groups and things, so we can all speak the same language, but we all have our biases.
Yeah, I mean, I think the way we're interpreting the current version of the NAPA legislation, it says and related dementias, and it defines what those are. But we have been given permission to include late, and it's been in the ADRD summit. And so as it is, even if we change the name or whatever, I think it's still included under, and it's been presented to the NAPA Council. And so I think in that sense, it's okay in terms of the funding can still be applied to this area, and it's in the milestones from the ADRD summit as well. So that's all in line for more research on this. And I agree completely with what Dr. Carlson said. I think, moving the science forward, especially if we can get to a point where we have something that might be a good biomarker, I think will really help to raise awareness. And, we're going to continue to, as a result of this, there'll be papers coming out from just this meeting, and then I hope it will stimulate lots of other research, and hopefully that will help to raise awareness. So I think, we're working on that, but that is part of what the discussion will be tomorrow, is about communication and dissemination. So not just for the scientists, but for the public and for providers.
Thank you, Nina. Greg has a comment too.
Yeah,
I do think that NAPA, we've talked about that the next 5 years, even though it says at the beginning Alzheimer's is meant to be ADRD, it needs to just say that up front so that we can make sure that people recognize the comorbid pathology.
So, I just wanted to chime in for those that weren't part of the meeting several years ago where the term late was developed, that it actually is descriptive. Limbic associated, age-related, TDP 43 encephalopathy. I think, a lot of the debate in the field was, is encephalopathy appropriate or not, which I thought was a silly argument. Part of the purpose was to come up with something catchy that would allow people to talk about it and bring it into not only common practice, and I actually have been using the term with my patients, and I actually find that they get it. And there's been a lot of press that's been out there, a lot of news articles, TV spots, radio spots. In Kentucky, people know what late is. We don't know much else, but we know what late is. So I just think it'd be very premature to start changing names all of a sudden. I think we need to really count on, it's not just us embracing this concept at a scientific level, it's the medical community, but it's also the patients, the media, the advocacy organizations, across the board. We all need to embrace this.
I'll just sort of add, hey, Brad, you go. You've been waiting, and you've got such a nice yellow hand.
Thanks, great to see you guys. Sorry I can't be there in person as well, but I'm glad we got into the discussion of the nomenclature, because on the one hand, I've been talking to patients about it too, Pete, and I think they do get it. And I usually put the encephalopathy in as a lowercase e, because most of them that I'm talking to about it don't have encephalopathy. So I think if we could just get rid of the encephalopathy and say you've got late, but that stands for limbic predominant age-related TDP-43 proteinopathy that tends to be, in the parts of the brain that are associated with memory, and you have a memory problem. And, we don't yet have a positive diagnostic test for that, but we're pretty sure to a moderate level of severity, or if we have, to a moderate level of confidence, or if we have a FDG PET, like I've been seeing too, that you guys really led the charge on, Dave and Hugo and others, we can be pretty confident, especially if we get CSF and rule out AD. And we should be getting those people into trials. So I think, one of the things you guys have done in this meeting and other forums finally is bring the AD and the FTD communities together. And I think that is something I really want to applaud right off the top, because there haven't been a lot of forums where we've been having this kind of crosstalk, and I think it's really badly needed. And I think there's so much good that can come of it. So I think if we could try to be inclusive of the people that have kind of pushed back against the encephalopathy piece, I think it would be a good thing. And I don't think that means you have to change the name. I just think you could make it a lowercase e or do something like that. And I do think we'd be really ripe for a natural history biomarker study. And, to me, that would really set the stage for the plans for larger scale clinical trials.
Yeah, I completely agree, and particularly agree about these sort of naturalistic studies. I think we'll have that opportunity now, again, as we're screening for people to enter treatment with lecanemab and donanemab. Those will be people that'll naturally fall into this category of people that we think are more likely to have underlying late than not. In terms of I think names obviously matter a lot, but I do think the clinical decision making is what Dave described earlier, which is, you start with a syndrome and then you put together other pieces of data that point you towards what you think the underlying biology is or pathology. And I do think while it's complicated in the community, I actually think that is a point of education that we need to continue to provide the community. I think you've actually, you and Ali and others have really tried to push that forward, is that, there's not this one-to-one matching between syndrome and pathology. And I think a lot of people in the community still think that. And I think it's incumbent on us, especially as we move towards, more molecularly specific types of therapies, that's a really important point of education.
I think,
I was going to say, we have another question online, but if that was a comment directly related.
yeah, going to that, I think we're comfortable with MCI due to Alzheimer's disease. I don't know why you can't do like amnestic disorder due to AD, amnestic disorder due to likely late or something like that, because that emphasizes that amnestic disorder is this umbrella and teaches people that there are different etiologies.
I think they're ready for it. The public's ready for it. If you take the time, they will understand. And I think one of the challenges before is that we didn't have all of these diseases known, one, and then two, we didn't have a ton of treatments to offer for different diseases. So talking about it didn't really matter to anybody because there was no change in what you were going to do clinically. But now that landscape's changing.
One other thing maybe related also to more observational data is I think as a research community, and again, maybe in terms of these pragmatic trials, we really do need to start to label, even when people have AD, what we think the likelihood is of late as a concomitant pathology using some of the tools that we've talked about today. Because I think only through that will we start to, one, learn how, whether those kinds of designations are correct, how they affect the natural history, and designing trials, again, for example, it might be nice to have an arm that's for like tau negative, A positive, tau negative, and A positive, tau positive. But the reality is even there, you might then need different length trials to, actually see an effect. And so I think we just really need, as a community to think about these cases and define them. And, the UDS will include late as one of the contributory pathologies that you can check off as either primary or secondary. And I think that's going to be really important for us to think carefully with. I was intrigued by Dave's data earlier where, your metric kind of found an intermediate level in AD. And so the question is, what do the ones that are higher on that level look like relative to those that are, lower on that level with regard to AD in terms of the likelihood of having underlying late? And, can we enrich in that kind of population and study them and then learn more through further refinement how to better, hone in on, which is the group that has the copathology?
Okay, we have questions piling up online, so let's go with, do you think certain environmental factors could drive disease progression in certain TDP-43 mediated late, microbiome being an important one, thinking about the heterogeneity?
I guess the short answer is yes, possibly. It wouldn't surprise me. I mean, again, I think, when we often think of all these sort of environmental factors, I think there's, a couple of potential pathways, and sort of the two broad ones I would say are environmental factors that either influence the underlying biology that you're looking at, which in this case would be TDP-43, and the other major pathway is how does that influence your resilience to that pathology. So, people tolerate different amounts of pathology and don't necessarily become symptomatic to the same degree. And so I think those factors are clearly important on that basis. But there likely are also, more direct biological mechanisms associated with a variety of the proteinopathies in terms of the environment.
Yeah, so I think it's a great question, and the answer I think is we don't know yet. But though we do have intriguing preliminary data in the ROS-MAP cohort about the dietary inflammatory index and the hippocampal inflammation, and hippocampal sclerosis. I'm sorry. So there, I think there's reason to believe just what you're saying is that it may increase your risk, some of these environmental factors, of actually going down one or another pathway.
And I will say that NIA recently got some money specifically to look at the exposome, broadly speaking, and we have some specific NOFOs, Notice of Funding Opportunity, we're supposed to call them now, out. So folks can ask me if you want to know about that. Okay, another question from Dietmar Thal. What would you like to include under the diagnosis late? Only pure late, or include AD plus late? How would you distinguish them as subtypes, or would you lump them? Distinguishing them will be useful for trials.
My own thinking is I think I wouldn't necessarily divide late in that way, but I think the context matters. So I would think about AD in the context of late and late alone, and that making those distinctions are important and have relevance to trials, and certainly have relevance to prognosis as well. In terms of the actual biology or the pathology itself and the staging, I think of them as being the same entity, or at least highly overlapping. And again, that these other contextual factors then maybe influence outcomes and progression. But Pete, you comment on this.
Wait, what were we? I was got side reeled in my head. What were we talking about?
So whether to separate, think of late as two different entities or subtypes if they're in the context of AD or not.
Culturally, it's sort of interesting. We used to hierarchicalize our neuropathological diagnoses. We used to say this person's primary thing was this, the secondary thing was this, and that was a very important thing back then. I never do that anymore, and I think it's wrong, and I think that the field has paradigmatically shifted away from that. We say this person has Brock stage 4 ADNC with A, B, C. We have late stage 2. We have this many microinfarcts. We do not have the crystal ball to know what exactly caused what clinical manifestation. We'd have no clue. That hierarchicalization is not possible. We can do a multivariable regression-based assessment of the attributable risk of each subtype of pathology in a population, but we cannot do that in a human being. So I think that we can say that Brock stage 6 ruined your whole day, but I tell you what, if you have the late stage 2 as well, you are down lower than if you just had the Brock stage 6. That's a fact. I'm sort of tempted whether or not to go into this nomenclature thing, but I'm going to let it sit for now.
But I think the question was about clinical, right? And so here's what I
It is, but I think that the clinical, Dietmar, if you have a suspicion of AD and late, that's a different clinical diagnosis from a suspicion of AD alone or late alone. It has implications, I think, towards both the therapeutic response of the individual and the prognosis of the individual. It has manifestations from the genetic upstream elements, and it has a lot of implications, I think, about your ultimate fate. And so I think that if we have a nomenclature that ignores this complexity, we're not doing ourselves or our field any favors. The fact is, we have had a silly attempt to dichotomize everything in our field. We have dichotomized clinical as you are demented or you are not demented. It turns out that whenever you look in a community-based cohort, most people are in between. Most people are in between, and that dementia line is different for different clinicians, and it's different, and it's sort of artificial. And the AD yes, no, dementia yes, no thing, it's done. We need to be through with it. And the fact is that if you want simplicity, going to the most complex system in the history of the universe, which is the aged human brain, is not the place to go. We have to grapple with the complexity. And my analogy is cancer research in the 1990s. They used to just nuke every cancer with a poison that they thought was going to kill the cancer faster than the person. Sometimes it worked, sometimes it didn't matter because cancer stinks. Well, but they then started to grapple with the complexity of the pathways that cause and contribute to cancer. And by so doing, developed therapies that were more specific, bent the curve of survival better, and were more tailored to the persons and the complexity of the diseases that were underlying. We are now starting down that pathway with a absolutely good biological that affects A-beta amyloidosis, which I think should provide us with a linchpin to get progress towards that other 70%. And to grapple with that complexity, deal with it, and deal with the fact. So no, Dietmar, you should not put that complexity under the rug, deal with the complexity, and that's what our success will come from.
So I interpreted the question the way Dave did also. And my answer to the question would be that right now I do think separating AD, TDP, or AD late, and then AD is a worthwhile thing to do from a therapeutic standpoint, and a diagnostic and a prognostic
standpoint. And that is what
standpoint.
we do clinically. We do that, but I wanted to have a slight caveat because what people may think is pure late, they probably won't be thinking of A pos T negative as pure late, but it still can be. So they might be thinking that that's AD with late. And the criteria that I presented and like the data really does indicate A pos T negative can still be pure late. And then when you are dealing with pure AD, that's certainly much more common when the medial temporal lobe is not involved in the syndrome clinically and on imaging. And those are young people with Alzheimer's disease predominantly. And so those are the two extremes, going all the way from the limbic system to heteromodal association cortices. Amnestic predominant, dysexecutive predominant. That's the extreme. Now, where you talk about mixed AD and late, in between. Right. And yeah, that exists.
So I think the answer is, right now we're still at the categorization stage. And it does, even though I understand exactly what you're saying, and sometimes determining if someone has dementia can be difficult, it still helps to bin people into these categories from a diagnostic standpoint.
Okay, we only have 11 minutes left, and I have a couple more questions. Some of them are going back to the clinical trials session online. So a question for Rema, which I think Dr. Boyle may also want to answer. Could it be that the neuropsych test variability is in part related to the association of AD with late at that age?
Yeah, I was just going to ask Dr. Boyle if she wanted to take this, because I know she talked about this exact question.
I don't know. That's a hard one. I think, it's possible. I mean, we don't know when these things are going on at the same time. We don't know exactly how that affects variability in cognitive testing. So I suppose it's possible. And certainly we do see, as everyone here knows, when two things are happening, late and AD in particular, are happening together, there's worse performance. We have looked at variability with different pathologies in the past, particularly Lewy bodies. That's something we could look at here in this setting to see, you know, what that actually looks like in the data. I think it's possible. I don't really know from the data that I can answer that well, though. Julia,
enough.
do you have any other? Yeah.
And I would just, I would also just say that it may not be late versus AD from the data that you just showed, Dr. Boyle. It might be age, some of these other confounds that might be driving these differences. And that once you account for those, and it just happens that late patients and our participants are going to be older. And so depending on the proportional representation in studies, you may have to account for the variability across the different confounds, like age, ApoE, maybe tau levels, whatever it might be.
And I think Patricia was saying, we would do that, like we did with the Lewy body, to show whether or not there's variability separate from those other compounds.
And I think
Absolutely.
you need samples that are fairly either representative, and you have to be able to validate this in other independent samples so that you can actually see what these sample biases are as well. So
I think the other thing to keep in mind is, and this has been brought up multiple times today, the older people with late also have a high rate of Lewy bodies and a high rate of vascular disease. So there's variability is coming in from multiple levels.
another question, I think this might be for Dr. Rissman. There was a paper presenting RT quick analysis in 2020 of TDP43 from CSF that's a lot of acronyms, CSF in an FTD cohort. Has anyone in this group attempted this assay in a population suspected of late neuropath changes?
I don't think so. So RT-Quick is a very specific technique, and it can't just be done by everyone. So I think the majority of those results, although interesting, have not been replicated. Yeah.
Thank you.
The recombinant protein is very hard to work with, so that's a very hard assay to do. Yeah,
it is.
Dr. Duggar, you have a question?
It's more of a thought-provoking thing for everybody for maybe tomorrow's discussion. And thinking about some of the operational aspects that people were saying. So you have a definition of something, and then you have all these downstream effects. You got to advocate it, you got to have legislation. So something to think about is when we come up with these terminologies, and I know there's a lot of stuff being done in parallel with other diseases in this realm, is how should we set like certain timelines for how frequently we should update things. This is done for DMS5, this is done for WHO criteria. A lot of other realms do this. And so just thinking of trying to optimize resources and task force to move forward and have kind of like this checklist workflow, because this late group is amazing, right? This is a really great kind of thing of thought making of the minds and everything, but you see these sprinklings like Dr. Kawas was like, oh, hey, what about vascular, right? And there's things coming down the road. So just thinking from that standpoint to maybe, it's more on a global thing. It's not necessarily TDP 43 related, but you think, what is it? The UDS form, we're just getting now to the 4, right? But now it's like, oh, wait, do we need to add more things? Or like Pete, you presented, or Dr. Nelson, you presented about, hey, can we add this module for late? In the new neuropath form that was just version 11, so should it be 11.1? So these are just things to think about from an operational standpoint,
I'll just say as a general principle, I think we should revisit, anything that we develop now, in the near future, because the field is changing, so quickly, and the sort of panoply of biomarkers and other tools we have is changing so quickly. And again, even from a therapeutic standpoint, that's going to really influence a lot of thinking in the years ahead. So it would be, I think we had a conversation earlier that if we're still, whatever we say today, if we're still saying it 10 years from now, I think we probably didn't do our job well enough, because we should be modifying it.
Well,
So is
throughout
that
the
a
day, we've had
5-year,
a lot of kudos online from lots of different people. Last one was Dr. Kukel, who just said, really great meeting, great presentations and discussions, and thank you, everyone. So I think we can end on that note, unless you'd like to make some closing remarks.
I don't need to make closing remarks other than that, I think it was a really robust set of talks and conversations today. And I do feel like in some sense we've made progress in moving forward and I think developing a degree of confidence and comfort around beginning to move this condition into the clinical realm, and also I think provided a lot of motivation for why I think it's important for us to do that. So thank you everyone here for participating, and look forward to continued conversations. And thanks, Linda, again for for being supportive.
So I think many of you are aware there is a dinner at Summer House. I think we actually moved the reservation till 7 p.m. so that everybody has some time to get back to the hotel if they need to. But it should be within walking distance of the hotel. Very close.
Yes, it is. It's very
Yes,
close.
I think it's like a half a block away. Erica told me, so that's great. Welcome, though. Please come. The dinner restaurant.
Not
From the
here.
hotel.
Hotel.
Hey, Dwayne.
Yes, sir.